Active transport occurs when molecules move across the cell membrane. The molecules move in the direction against a gradient, which is most often a polar repulsion or other type of resistance. During active transport, transmembrane proteins selectively choose which substances are allowed to cross the membrane. The two types of this process are primary active transport and secondary active transport. During primary active transport, aka direct active transport, the proteins act as pumps that use chemical energy in the form of ATP. The process directly uses metabolic energy to move molecules across the membrane. Most of the enzymes that are involved in this type of transport are called transmembrane ATPases. Other sources of energy are redox energy and photon energy. An example of this type using redox energy is the mitochondrial electron transport chain that uses the reduction energy of NADH to move protons across the inner mitochondrial membrane against the concentration gradient. An example of primary active transport using light energy are the proteins involved in photosynthesis that use the energy from light to create a proton gradient across the thylakoid membrane and to create reduction power in the form of NADPH. On the other hand, secondary active transport requires potential energy that usually comes from the manipulation of an electrochemical gradient. This process involves proteins that form pores to create channels across the cell membrane. Secondary active transport is also known as coupled transport. This involves energy used to transport molecules across a membrane, whereas primary active transport does not involve direct coupling of ATP. It instead relies on the electrochemical potential difference caused by ions pumping in and out of the cell. The word lysosome comes from Greek words lysis, meaning to loosen, and soma, meaning body. It is a membrane-bound cell organelle that is found in most animals. Lysosomes are spherical vesicles that contain hydrolytic enzymes that break down all sorts of biomolecules, including proteins, nucleic acids, carbohydrates, lipids, and debris. Lysosomes can be described as the waste disposal system of the cell. They digest unwanted materials in the cytoplasm, both from outside and inside the cell. That is why they can be referred to as suicide bags or suicide sacs of the cell. Lysosomes are also responsible for homeostasis within the cell. They are involved in secretion, plasma membrane repair, cell signaling, and energy metabolism. Enzymes of the lysosomes are synthesized in the rough ER and released from the Golgi apparatus in small vesicles, which eventually fuse with acidic vesicles called endosomes. They then become full lysosomes. The endomembrane system is composed of the different membranes that are located in the cytoplasm inside a eukaryotic cell. These membranes separate the cell into functional and structural compartments, or organelles. In eukaryotes, the organelles of the endomembrane system include the nuclear membrane, the endoplasmic reticulum, the Golgi apparatus, lysosomes, vesicles, endosomes, and the cell membrane. The system is defined as a set of membranes that form a single functioning unit that is either connected directly or by exchanging material through vesicle transport. It is important to remember that the endomembrane system does not include the membranes of mitochondria or chloroplasts. The nuclear membrane contains two lipid bilayers that involve the objects in the nucleus. The ER is a synthesis and transport organelle that extends into the cytoplasm. The Golgi apparatus is a chain of several compartments where molecules are packaged in order to be delivered to other components inside the cell. A vesicle is a sac in the membrane that stores or transports substances. The organelles within the endomembrane system are related through either direct contact or by transfer of membrane segments as vesicles. One characteristic that all the membranes share is that they all contain a lipid bilayer with proteins across or on either side of them. Centrioles are cylindrical cell structures that are composed mainly of a protein called tubulin, which are found in most animal cells. Most centrioles are made of 9 sets of microtubule triplets that make up a cylinder shape. They're involved in the organization of the mitotic spindle and in the completion of cytokinesis and can self-replicate during cell division. Centrioles play a key role of centrosomes, which are involved in organizing microtubules within the cytoplasm. When two centrioles are located next to each other, they are usually at right angles. They are found in pairs and move towards opposite ends of the poles of the nucleus during cell division. During cell division, 4 centrioles will appear. One pair will move in each direction. Interphase is when the cell is at rest. Then, during cell division, the centrioles duplicate. During prophase, the centrioles move to the opposite ends of the nucleus and a mitotic spindle of threads begins to appear. The threads then connect to the chromosomes. During anaphase, the chromosomes split and pull towards each centriole. In telophase, the entire cell begins to split, and the chromosomes begin to unravel and new nuclear envelopes begin to appear.
The absence or presence of a nucleus determines the type of cell as a whole. A eukaryotic cell has a nucleus, whereas a prokaryotic cell does not. Having a nucleus also indicates the cell's ability to create cell membranes. The nucleus, a double-bound membrane, is comprised of 2 lipid bilayers. This makes up the nuclear envelope. In order to enter the nucleus, you must first go through what are called the nuclear pores. These pores are highly regulated in order for only certain macromolecules to pass through. Inside the nucleus is the nucleolus, the site of active transcription and making of RNA. The priority of the nucleus is to protect the cell's DNA. In the resting state of a nucleus, chromatin, which makes up chromosomes, are present. These are made up of DNA, RNA, and nuclear proteins. Peroxisomes are small membrane-enclosed organelles that contain enzymes involved in a variety of metabolic reactions, including several aspects of energy metabolism. Peroxisomes contain a variety of enzymes which primarily function together to rid the cell of toxic substances, and in particular, hydrogen peroxide, a common byproduct of cellular metabolism. These organelles contain enzymes that convert the hydrogen peroxide to water, rendering the potentially toxic substance safe for release back into the cell. Some types of peroxisomes, such as those in liver cells, detoxify alcohol and other harmful compounds by transferring the hydrogen item from the poisons to molecules of oxygen. This process is called oxidation. Peroxisomes usually use significant amounts of oxygen in order to carry out their activities. They're also similar to appearance to lysosomes, another type of microbody, but the two have very different origins. Whereas lysosomes are generally formed in the Golgi complex, peroxisomes self-replicate. However, they do not contain their own internal DNA molecules. The cytoskeleton is a structure that helps cells maintain their shape and internal organization. It also provides mechanical support that enables cells to carry out essential functions like division and movement. The cytoskeleton of eukaryotic cells is made of filamentous proteins and provides mechanical support to the cell and its cytoplasmic constituents. All cytoskeletons consist of 3 major classes of elements that differ in size and in protein composition. Microtubules are the largest type of filament, with a diameter of about 25 nanometers. These are composed of a protein called tubulin. Actin filaments are the smallest type, with a diameter of only about 6 nanometers, and are made up of a protein called actin. Intermediate filaments are middle-sized, with a diameter of about 10 nanometers. Unlike actin filaments and microtubules, intermediate filaments are constructed from a number of different subunit proteins. Microtubules tend to grow out from the centrosome to the plasma membrane. They radiate out from the centrosome to provide the basic organization of the cytoplasm. including the positioning of organelles. Actin filaments are also involved in cytokinesis and cell movement. Intermediate filaments come in several types, but they are generally strong and rope-like. Their functions are primarily mechanical, and intermediate filaments are less dynamic than actin filaments or microtubules. Mitochondria are unusual organelles. They act as the power plants of the cell, are surrounded by two membranes, and have their own genome. They also divide independently of the cell in which they reside, meaning mitochondrial replication is not coupled to cell division. Mitochondria contain two major membranes. The outer mitochondrial membrane fully surrounds the inner membrane, with a small intermembrane space in between. The outer membrane has many protein-based pores that are big enough to allow the passage of ions and molecules as large as a small protein. In contrast, the inner membrane has much more restricted permeability, much like the plasma membrane of a cell. The inner membrane is also loaded with proteins involved in electron transport and ATP synthesis. This membrane surrounds the mitochondrial matrix, where the citric acid cycle produces the electrons that travel from one protein complex to the next in the inner membrane. At the end of this electron transport chain, the final electron acceptor is oxygen, and this ultimately forms water, H2O. At the same time, the electron transport chain produces ATP. During electron transport, the participating protein complexes push protons from the matrix out to the intermembrane space. This creates a concentration gradient of protons that another protein complex, called ATP synthase, uses to power synthesis of the energy carrier molecule, ATP. There is compelling evidence that mitochondria was once a primitive bacterial cell. This evidence is described as the endosymbiotic theory. Symbiosis occurs when two different species benefit from living and working together. When one organism lives inside the other, it is called endosymbiosis. The endosymbiotic theory describes how a large host cell and an ingested bacteria could easily become dependent on one another for survival, resulting in a permanent relationship. Over a million years of evolution, mitochondria have become more specialized, and today they cannot live outside the cell. Mitochondria also have similarities to bacteria cells. It has its own DNA, which is separate from the DNA found in the nucleus of the cell. The mitochondria's organelles use their DNA to produce many proteins and enzymes required for function. A mitochondria also has a double membrane that surrounds it. Mitochondria also reproduce like bacteria, replicating their own DNA and directing their own division.
Secretory proteins are made on ribosomes bound for the rough ER. The proteins move through the endomembrane system and are dispatched from the transphase of the Golgi apparatus in transport vesicles. These vesicles move through the cytoplasm and fuse with the plasma membrane. When they fuse with the plasma membrane, the protein is released to the outside of the cell in a process called exocytosis. The membrane proteins have a similar pathway, but differ when they reach the plasma membrane. When they reach a plasma membrane in transport vesicles, the protein is not released to the outside of the cell. It remains fused with the plasma membrane. Facilitated transport is a form of passive transport. It happens through a protein channel or pore. Each channel or pore is specific to the chemical passing through it. It transports the molecules from high concentration to low. The cell membrane's function is to protect the cytoplasm from the exterior environment. It is a selectively permeable membrane, which means that it regulates what goes in and out of the cell. The structure of the cell membrane contains a phospholipid bilayer. The phospholipid bilayer consists of two layers of phospholipids that have the hydrophilic head on the outside and the hydrophobic tail on the inside. The structure can also contain peripheral proteins or transmembrane proteins. The fluid mosaic model was made by S.J. Singer and G.L. Nicholson in 1972. However, there has been many modifications and additions made to it since then. The model explains the observations made in regards to the structure of the cell membrane. The mosaic part in the name refers to the proteins made up of the cell membrane. They can be either peripheral or integral. The fluid part is in regards to the phospholipid bilayer. The phospholipids depend on the temperature and the liquid composition. The bilayer may become more of a gel or even a solid, or it can be a liquid. The bilayer is usually liquid, which helps with the diffusion of proteins. Membrane proteins, like all other proteins, are made in the ribosomes. These proteins have many different functions. Membrane receptor proteins pass signals between the internal and external environments of a cell. Transport proteins move molecules and ions across the membrane. Membrane enzymes perform with reactants. Cell adhesion molecules bind with other cells and their surroundings. There are two types of membrane proteins. There are integral membrane proteins, which are permanently attached to the membrane and span across the membrane at least once. There are also peripheral membrane proteins that are temporary and attached to the bilayer or an integral protein.